Hello, Bio30. Welcome to the second video on this one is on cell division and interphase. It's the second video in our molecular genetics unit. So today we're going to talk about the cell cycle and some other very important key vocabulary words. The point of today, so how do you know you've actually accomplished what you need to accomplish, is being able to tell me the different parts of the cell cycle, such as G1, G2, S phase, and interphase. There's going to be a pie chart at the end. I call it a pie chart. It's kind of just more of a diagram that you should be able to draw for me, as well as there's going to be a lot of key vocab words right at the beginning. Those are going to be for the entire unit. There's going to be lots of vocab, so please pay attention, get them in your notes, and let's start using them as working definitions as we go through this chapter. So the first thing we're going to talk about is a parent cell and a daughter cell. So here's a parent cell. We're going to call it my original cell. Okay, it's what I'm starting with. When that cell divides to replace itself, maybe it's got some damage or maybe it's coming to the end of its cell life, it's going to split and create new cells. The dividing of that cell then becomes daughter cells. So parent cells are the original. Daughter cells are what are created from it. You may recognize this kind of small diagram from maybe Science 9 when we talk about asexual reproduction, or maybe back when we talked about mitosis then. It's just very simple, original to new. Here we have two words that you may remember from yesterday's lesson, chromosomes and chromatin. I talked a lot about chromatin looking a lot like a spaghetti-like structure, and chromosomes being that condensed version of it. So chromosomes are condensed DNA. They're taken from the long strands and really wound tightly up so that it's condensed. Chromatin is the long kind of spaghetti stuff, like I said, the long intertwined strands. There's also a piece on a chromosome called a centromere, which is right in the center. This is going to become really important when we start talking about mitosis and meiosis. So if I was to diagram what those kind of look like, you can see the cloud of scribbles kind of looks like chromatin, long intertwined strands. Then right here, this little X-like structure, or what looks like it could be a very weird shaped X, is actually a chromosome. This is showing the chromosome condensed down, tightly wound up. Okay, and this is showing a replicated chromosome. So it has two parts to it that are identical, held together by the centromere. So here's that centromere in the center. Now, when I'm talking about a chromosome in this form, if I'm talking about the individual sides of them, I refer to them as sister chromatids. So them together, sister chromatids. Here's where it gets a bit confusing. It may make more sense, or hopefully will make more sense when we get a little further on. This lovely guy right here, also a chromosome, okay? He's just not replicated yet. He hasn't doubled. He's just hanging out all by himself as just a condensed chromosome, all right? When we get to mitosis and meiosis, this is going to become important. So some other key vocab words that we're going to talk about is the word homologous chromosome. You're going to see this come up again later. So what it means is that all of our body has 46 chromosomes. And when we talk about them, we actually talk about them in pairs, because there's the ones that pair up because one comes from mom and one comes from dad. And then they're read and expressed in your body. So each one has its own pair. So women have 23 homologous pairs. Men have 22 homologous pairs with one sex pair chromosomes because they have XY. The Y chromosome is a little bit smaller than the X chromosome. Women have two X's. We also have what's called genes. Now, genes, like I said, are actually really important. You may remember me briefly talking about them yesterday. They're actually what expresses key information that makes up you. So there are areas of the DNA that contain specific genetic information that tells us important stories about you. Now, if I was to look at the movement through this, here's another just quick diagramming through how chromatin condenses to chromosomes and some keywords. So if we're looking at my cell, which is that big circle, I have a nucleus. This is my nucleus. And inside my nucleus, I have all this DNA. And this DNA is now replicating. You can see the two different colors. But currently it's in chromatin, the long strands. Now, what does DNA look like? Maybe from yesterday's video, remember you got this like backbone with phosphates and sugars, nitrogenous bases linking up in the ladder rung. But within that DNA strand, you also have specific areas that are genes coding for information that is you. If we pull it out and we say it's condensed, we condense down into one chromosome or into a replicated chromosome. And again, these two together we refer to as sister chromatids. Now you might want to draw this picture somewhere just to help you visually see the vocab words. It might be helpful. So the last couple vocab words are diploid, haploid, and polyploid. Now ploidy, Ploidy, just means the number of chromosomes in your body. So diploid, the di, so di, 2. How many? So we're 46. 2n equals 46. Haploid is n equals 23. So you can kind of see how 2n means the full amount, n means the half amount. Some animals, or some organisms, I should say, have more than that. Okay, they have more sets. They have more than 2 sets. So something like strawberries, for example, is 4n. At this point, I usually would like to show you a variety of different animals with different ploidies, because obviously we have 46, but other species have so many different types. Kangaroos, I think, have 16 fruit flies, or 8 or something like that. You can go on and look all over and you can find out what is the ploidy of most organisms. So at this point, if you want to stop, rewatch some vocab, make sure you have it, you can. I'm going to move on to the cell cycle. So what is the cell cycle? It is essentially the lifespan of the cell. All right, we go through our own life cycle from baby to adult to elderly, and then obviously pass on. The cell has a similar thing. So depending on the cell will depend on how fast they regenerate. Blood cells, skin cells replace quite frequently. You cut yourself, you lose a little bit of blood, it scabs over and it heals. You have to replenish those cells quickly. Something like nerve cells, if you think back to our very first chapter, nerve cells divide very slowly. In fact, some nerve cells can never be fixed, but the ones that can, the cells that replace them, do it very, very, very slowly as they divide very infrequently. Your body is a whole new body every 7 years. All the cells in your body should have divided at some point in time within the last 7 years. So every 7 years, all new cells. So what does this look like? We refer to a single cell cycle as one division to the next. And you're going to see what that looks like as I move forward. So there's usually 2 phases within a cell cycle. The first phase is interphase, and then is cell division. All right, so we're going to talk about what does the big word interphase mean, and then what is cell division. So interphase includes these 3 very key pieces, G1, S, and G2. Now, for your purposes, I do not care if you write down what happens in G1 and G2. Just know that G1 happens, then DNA replication in S phase, and then G2. All right? Nothing else needs to matter. Just know the order, G1, S, G2, and also know that S phase is DNA replication, because that's what we're going to talk about in the next few days. So what is DNA replication? Well, essentially, it's when each chromosome's replicating, and it can then has 2 sister chromatids. So it's in that X form. And that's going to become important when we start talking about mitosis and meiosis. G1 and G2 are essentially about cell growth, increasing energy, and making sure we have enough energy for cell division. So if I was to look at the cell cycle and label it as a very simplified diagram, I would refer to that entire large section there from this little tiny bit of the triangle all the way around. That is the entirety of interphase. Interphase is that entire part followed with cell division. So those are the 2 parts of the cell cycle. The majority of the time the cell spends in G1. Which again, I said you don't need to know what it is, but just know generally, I'm not going to ask you questions about it, but it's just rapid growth and metabolizing. S phase is followed right here. This is the one that I want you to know that it means DNA replication. Lastly, it goes through G2, which is just again preparing for division. So this cell will spend a large portion of its time in interphase, and then we'll go through cell division. Once it ends cell division, it's done its first round of its cell cycle, if that makes sense. So ladies and gentlemen, that is the cell cycle. Please ask me questions if you need to. You know how to get a hold of me, and I hope you have a great day.